Let's talk about protein function, the function of proteins. And I can read your mind. Some of you basically want to know, do I have to be able to draw this from the exam? Do I have to know the structure of this? No, you do not have to. Yes, you have to draw it, okay? And don't forget the little carbon dioxide molecules, the little dots over here. Otherwise, we're going to really take a lot of points off. Okay, so what you see over here is Rubisco. That is an enzyme. That's an enzyme, it's a Rubisco, and there are a few things here that look quite familiar. Do you already see these little spirals over here? These are known as alpha helices. Okay, and do you see these green arrows? I don't know, over here, yeah, you see there's an arrow over here pointing downwards. I'm down here, okay? These are known as the beta sheets. And the whole thing over here is an enzyme, and the name of this enzyme is ribulose 1,5-bisphosphate carboxylase oxygenase. That's a really cool name if you want to impress your parents. I'd suggest that you essentially at least memorize this name over here. And because it's a little bit like a tongue twister, then the scientists said, okay, now I think we're getting a little too complicated here. Let's invent a simpler name. Let's just call it Rubisco. Okay? So if you actually know Rubisco, that's fine. Okay? And this is basically the most common enzyme that can be found in nature, and it is responsible in photosynthesis for fixing carbon dioxide. So that is actually the enzyme which catches the carbon dioxide. I think these little red dots over here are supposed to represent the carbon dioxide, and basically to make glucose. So plants use this quite a lot. So that's nice also as a picture, because it also shows you that the two secondary structure motifs, namely the alpha helix and the beta sheet, because in the previous video, or the video with the hemoglobin, we did not see the beta sheet. And I just had to tell you that these are arrows, and if you look very carefully, you can actually see that these are small arrows, okay? So that is basically the first example of a protein function. So proteins can be enzymes, example Rubisco, and it's used for photosynthesis. So, second example is the hormone insulin. So, the category is a hormone. An example would be insulin. It causes cells to absorb glucose. And so, the pancreas releases insulin as a hormone, and basically this insulin then tells the cells to absorb more glucose. And I also have a picture of that. Here it is. Here is insulin. And, okay, over here I still have the drawing with the alpha helix, which is not correct. Okay, so I'm going to remove this. It's the previous drawing. And again, you see over here that there are these spiral-shaped alpha helices. Okay, and that's basically, yeah, a little bit simpler than the Rubisco enzyme, but it's still made of amino acids. It's still a chain of amino acids. It still has a three-dimensional shape. Okay, so that's the second example of a protein. And then there are these white parts. You might wonder what these white parts are. Well, these white parts are also polypeptides. It's not polypeptide, also a chain of amino acids, but they are not in a spiral. They're not in an alpha helix or in a beta sheet. So for this reason, they just left them white. And the last one are immunoglobulins, or also known as antibodies, and they bind against pathogens. And pathogens are disease-causing organisms, or can also be viruses. So let's have a look at one of these immunoglobulins, and that's how it looks like. Okay, and you see there are a lot of these beta sheets in here, a lot of these arrows. A few alpha helices over here, but mostly beta sheets. And sometimes a simplified form of drawing an immunoglobulin is a Y-shaped. Yeah. So sometimes in books, and I will also use this if I want to draw an antibody, I'm simply going to draw a Y-shaped structure because it has this Y shape. Okay, so that is another example of a protein function, and there is indeed the next foil, but I have to erase it. Well, I'll just go on to the next foil. Yes, and now I'm not giving you any pictures. Proteins can also be pigments. In this case, for example, rhodopsin. And you find this in the eye. It makes the rod cells in the retina, in the eye, light sensitive. So the rod cells are those cells which are not color sensitive, or in other ways, they cannot distinguish color. They're sensitive to all colors at the same time, and therefore you cannot distinguish color because they respond to all colors of the light equally. In any case, there's a protein in these photoreceptors in the retina in the eye to make them light sensitive. There's a structural protein, namely collagen, and these are fibers which prevent skin from tearing, gives bone, tendons, ligaments their strength. Essentially, collagen prevents that you basically fall apart. It holds your, it connects your skin to the tissue beneath it, and so on, okay? Something that we actually don't think of a lot is essentially we have to be held together. Our organs have to be held together. And collagen is responsible for doing that. And last, another example, spider silk is also a structural protein, and it makes spiders' webs become stronger and when stretched. So you stretch the spider web, and actually, as a matter of fact, it even becomes stronger. So essentially, what's the point of this table? The point of this table is simply to show you that even though all of these proteins are made of the same 20 amino acids, simply connected differently, the function and the structure can be vastly different. The analogy that I always use is the following. I mean, you have only 26 letters in the alphabet, but essentially, how many different words or sentences or even books can you write? It is not unlimited. There is a theoretical limit, but it's so huge that you can almost consider it unlimited. So is it possible to make proteins that don't do anything at all, that are completely pointless, senseless? Of course you can do that. You can also write words which are completely pointless and senseless, which don't make sense and which don't exist. Of course you can do that. But essentially, the proteins that the cells, living things make, of course, they have a function, and you have already seen over here that the functions can be extremely diverse and quite different. Okay, so what should you know? You have to know the different categories of protein functions over here. And if I go back over here, these are different categories and also some examples. But of course, there are many more examples. I mean, there are many, many more examples of enzymes. Okay, and there are also different types of immunoglobulins around, and so on. So the idea here is simply to give you an overview of what is essentially possible. And the second thing, it should also justify a little bit why we spend so much time on proteins. Because you see now that essentially the proteins are the things in our body, in our cells, which actually do the work. And they do a lot of different things. Okay, so that is basically, I hope I've given you now a little bit the big picture of why an understanding of protein chemistry is so important to understand how things work in biology.